السلام عليكم ورحمة الله وبركاته. Lucas Biopharmaceutical Biosimilar by Jamal Almaamari. History of biotechnology. Biotechnology is in some ways as old as human history. Our ancestors harnessed living organisms to make bread, cloth, and cheese and wine. It was just during the early 20th century when the term biotechnology came into use. The term was coined in 1917 by Karl Ilke, a Hungarian engineer and a professor who described a technology based on converting raw material into some useful product. At that time, the newly categorized field was focused on the food production, addressing such issues as malnutrition and famine. Falson expanded its focus to medical uses led by the 1940 introduction of the penicillin made through a defermentation process greatly impacted countries' lives over a half century ago. Today, biologic medicines are making significant impact on the lives of patients with serious illness throughout the world, hold promise to cure diseases like cancer, Alzheimer, multiple sclerosis, arthritis, and cardiovascular disorder. The definition of, or the standard definition of biotechnology, any technological application that uses biological system, living organism, or derivatives thereof to make or modify products and processes of specific use. Biopharmaceutical drug created by many biotechnology, especially genetic engineering, primarily combine DNA, protein, and monoclonal antibody, typically derived from living organisms like animal cell, bacteria, viruses, and yeast. Therapeutic proteins, cytokine, hormone, clothing factor, insulin, DNA, vaccine, monoclonal antibodies. New experimental modalities such as gene therapy, stem cell therapy, and RNA viruses. This example of biosimilar compared with the reference product name and general or official generic name INN name, international non-proprietary name. Biological medicines are currently prescribed to treat a wide variety of diseases, including blood condition, leukoneutropenia, some cancers, colon, breast, and some diseases in immune systems, other rheumatoid arthritis, psoriasis, and Crohn's disease, and some neurological disorders, multiple sclerosis. More than 400 biologics are in the clinical trials. These include therapy for cancer, Alzheimer, heart disease, diabetes, HIV, AIDS, and autoimmune disease or disorder. Represent a fast growing segment of the pharmaceutical market, constituting 32% of product in the development and 7.5% of marketing products expected to grow exponentially at more than 20% per year. By 2016, 7 of top 10 pharmaceutical worldwide will be biologics, potential to reach up to 50% share in global pharmaceutical market in the next few years. Manufacturing of biopharmaceutical develop host cell. This is the step of develop host cell from identify the human DNA sequence and isolate of DNA, select vector, insert the gene to genomic or host, modification of the cell, combine DNA or technology, the exact DNA sequence and type of host cell used will significantly influence the characteristic of the product. This is the major step involved. Manufacturing of biopharmaceutical from start by modifying the selected cell, growing cell line, growing a large number, qualitative team, and separating a protein from the cell, and finally by purifying the collected protein. The differences in manufacture of conventional drugs and biologic. Here, small molecule drugs represent conventional drugs, small molecule drugs, low molecular weight, made by adding or mixing together a known chemical reagent and series of controlling and predictable chemical reaction. Product techniques usually same as for inhibitor production process, highly standardized. Contamination are consistent and quantifiable, while biopharmaceutical strong relationship between manufacturing process and biopharmaceuticals and characteristic of the final product manufacturing biology is more complex, comparable with the conventional drugs. A high level of protein is required to produce a consistent product time after time. This is the summary of a step of manufacturing or differential between conventional drugs and biosimilar drugs to assure high quality and consistency in the final product. Production process require a high level of monitoring and testing throughout the process. Biologic drugs typically has around 250 in-process testing during manufacturing, comparable to around 50 tests for small molecule drugs or conventional drugs. Biosimilars. What are the biosimilars? Legally approved subsequent version of innovator biopharmaceutical products made by a different sponsor for patent and exclusively expiry of innovator products. Because of structural and manufacturing complexities, these biological products are considered are similar but not generic equivalent of innovator biopharmaceutical. The definition and interpretation of biosimilar products. This definition by WHO definition similar to an already licensed different biotherapeutic product in terms of quality, safety, and efficacy. While United States Food Drug Administration definition, the biosimilar is highly similar to the reference product without clinically meaningful differences in safety, purity, and potency. Based on this different definition, there are three dimensions in the definition. In the definition of the biosimilar product, number one, or it should be biologic product. If the reference product should be a ready licensed biological product, the demonstration of highly similarity in safety, quality, efficacy, and necessary. The similarity should be demonstrated using a set of comprehensive comparability exercise at the quantity, non-clinical, and clinical level. High unit cost of biologic has resulted in patient concern about continuous across the potentially effective therapy. Since the expiration of patents for a number of blockbuster biological hazards shared in an area of the subsequent production of biosimilar product contribute to increase access to this product at an affordable price. The global scenario in 2010 scales of biological risks 100 billion worldwide, with the top uh, 12 biologic generating 30 billion dollars. By 2015, biologic response for $20 billion in annual sales will go overbeating. Global market for biosimilar was $311 million in 2010 and expected to increase to $2 and $2.5 billion in 2015. Problem statement: Biosimilars are not biological generic unique molecules which are supported by only limited clinical data at the time of approval. Concern regarding their efficacy, long-term safety, and immunogenicity. The generic drugs, chemically and therapeutically equivalent to the branded original molecular chemical drugs, both patents have expired. Identical to the original product, must country really have well-established scientific standard and legal mechanism for authorizing generic. Approval of generics in 1984 for United Food Drug Administration was authorized to approve generic drug product under the Hart-Coxman Act. When an innovator product is going off patent, pharmaceutical company file an abbreviated new drug application for approval of generic copies of innovator product. According to Food Drug Administration definition, the generic drugs product should be comparable to the reference drug product in the dosage form, strength, route of administration, quality, performance characteristic, and intended use. Authorized on the basis of demonstrating that they are the same in structure and bioequivalent to a profit product. Require evidence of comparable bioavailability, conduct of bioequivalent studies. Non-clinical and clinical are not usually required. Recognize it for some time that these regimes will not work at biological derived from drugs. Differences between generic and biosimilar. However, unlike structurally well-defined low molecular weight chemical drugs, biopharmaceutical are high molecular weight compounds with complex three-dimensional structure. For example, molecular weight of aspirin 100 Dalton, weight interferon beta is 19,000 Dalton. The differences the larger differences typically biologic drugs is 100 to 1000 times larger than small molecule chemical drugs. Possesses fragile three-dimensional structure as compared to well characterized one-dimensional structure of the chemical drugs. Difficult to define structure, small molecule drugs easy to reproduce, specified by mass spectroscopy and other techniques. Lack of appropriate investigation tools to define composite structure of larger protein. Complex manufacturing process. Manufacturer of biosimilar product will not have access to manufacturing process of innovator products, proprietary knowledge, possible to accurately the duplicate and protein product. Different manufacturing processes use different cells lines, protein sources, and extraction and purification techniques. Heterogeneity of biopharmaceuticals. Versatile cells line used to produce the protein have an impact on the gross structure of the protein. Such alteration might significantly impact receptor binding stability and pharmacokinetics and safety and immunogenic potential of therapeutic proteins. Onyx safety issue not observed with chemical generics. The issues of the issues concern with use of biosimilar efficacy issues, difference between bioactivity of the biosimilar and innovator products, safety issue, concern regarding immunogenicity and pharmacovigilance. Due to limited clinical database at the time of approval, vigorous pharmacovigilance is required. immunogenicity and adverse drug reaction, type of adverse event, and uh, 4. Substitution allows dispensing of generic drugs in place of prescribed innovator product. Rationale of generics, original drugs, anti-generic are identical and have the some therapeutic effect, reduce cost saving. Some substituent roles should be not applied, like decrease the safety of therapy, of course, therapeutic failure, uncontrolled substitute, and adverse event emerges after switching from the innovator product to the biosimilar with documentation. Event will not be associated to the specific product, or it will be described to a wrong product. uh, Naming and labeling, uh, generic adaptation of chemical medicines assigned to the same name, identical copy of the reference product, biosimilar required on INNs as the wall facilities, prescribing and dispensing biopharmaceutical price prices, pharmacovigilance, need for comprehensive labeling for biosimilar, including the derivation from innovation product and on activity and efficacy data, assist physician and pharmacist in making informed decision. Uh, Here the status of regulation for biosimilar globally, a strong need for regulation governing biosimilar implementation of the abbreviated licensure pathway for biological products presents challenge given the associated scientific and technical complexities. European Union has regulation in place for quite some time for approving biosimilar. United States have recently covered this under the respective act by bringing in the acceptable guideline for their evaluation and for regulation. World Health Organization guidelines, scientific basis for the evaluation and regulation of the biosimilar was discussed and agreement for developing WHO guidelines will record at the first WHO informal consultation and regulatory evaluation of therapeutic biological medicine product held in the Geneva 2007 year published guideline in evaluation of similar biologic products with detailed recommendation on clinical development in October 2009. Regulatory framework in European guideline on similar biological product containing biotechnology-derived proteins as active substance were adopted by European Medicine Agency in June 2006, issued product-specific biosimilar guideline in European Union. The first patent on biopharmaceutical expired in 2001, and first biosimilar medicine was approved by European Union in 2006. In 2018, the European biosimilar market generated revenues of approximately $172 million. Regulatory framework in the United States. United States Food and Drug Administration issued the draft guideline document are recent as 19th February 2012 on biosimilar product development under Biologic Price Competition and Innovation Act of 2019 based in sponsor providing structural composition and clinical similarity with an approved biologic. include important of extensive analytical and physiochemical biological characterization in demonstrating that proposed biosimilar product is highly similar to the reference product, notwithstanding minor differences in the clinical inactive components. Manufacturing process required before conducting a preclinical study. The preclinical submission stage include a complete description of molecular biology consideration, uh, like hostile factors, gene sequence, promoters, and post-translation modification, glycosylation, oxidation, diamidization, phosphorylation, uh, fermentation process development, and protein purification details should be provided, and product characterization like physiochemical properties, biological activity, immunochemical properties, purity and contamination strength and content. Preclinical study comparative in nature and designed to detect differences. Study design depend on the therapeutic index type, number of indication applied, conducted with the final formulation, dosage form, strength, route of administration. The profile period, they conduct the study statutory approval from respective institution or authority or committee or commit. Preclinical evaluation include pharmacodynamic study in vitro or vivo, toxicological studies, immunoresponse in animals, and clinical study include pharmacokinetic studies designed show taking full factor in the consideration half-life, linearity of pharmacokinetic parameters, endogenous levels, and other variation. Pharmacodynamic study, confirmatory safety and efficacy study, safety and immunogenicity data, extrapolation of efficacy and safety data to other indication, and market authorization application. And we need post-market data for similar biologics like pharmacovigilance, plain and adverse drug reaction reporting, post-marketing study, and archiving of this data. Thank you.